or feature in the life cycle of a plant. It is also an unique feature of the plant species to overcome unfavorable conditions. Even in agriculture, it has a lot of significance. I am Dr. Shubha, Associate Professor of Botany, Government First Grade College, Vijayanagara, Bengaluru, and I'll be speaking about dormancy today. In the previous video, I have discussed about photoperiodism and then vernalization. Dormancy in flowering plants is seen in the two kinds of propagules, the bud and the seed. You must have seen during winter all the leaves will fall, especially in the temperate countries where the temperature is very low, because the leaf buds are undergoing a period of rest. And even you must have seen whenever we harvest any plant or any product for that matter, agriculture produce, most of it will not germinate immediately after the harvest. It may take some time, and this is the resting period, and this is a very important feature of any plant that is called dormancy. Usually when the environmental conditions like moisture, temperature, humidity, etc. are favorable, seeds will germinate and buds will grow. But this may not happen in many cases. Seeds fail to germinate and buds may fail to sprout. This may be delayed for days or months or even years. That is, they are in a state of dormancy which may be due to internal conditions. What are the types of dormancy? And then before that, let us see about the definition of the dormancy. Dormancy is defined as any phase in the life cycle of a plant in which active growth is temporarily suspended. It is seen in two kinds of propagules, as I have already mentioned, that is the buds and seeds. Coming to the types, there are two types of dormancy, imposed dormancy and innate dormancy. Imposed dormancy occurs due to unfavorable conditions, and innate dormancy is due to the conditions within the dormant organ itself, that is innate or inbuilt in the system. In this kind, even if external conditions are favorable, growth will not occur. There are three different phases of dormancy, that is pre-dormancy, mid-dormancy and post-dormancy. And in pre-dormancy, it is the early phase during which organs may be induced to resume growth by various treatments. That is, this is the period before the proper dormancy starts, and in this case or during this period, if we treat the plant organ or the seed or bud with some chemicals or some things which can break the dormancy, it can be broken. The next thing is mid-dormancy. It is a state of full dormancy during which it is difficult to induce growth by changing external conditions. Next is post-dormancy. It is a state of gradual emergence from dormancy during which growth can be induced. First, let us consider bud dormancy. Buds produced in plants to bring about vegetative propagation. That is, the buds are very important from the point of view of propagation of plants, and these buds help in the multiplication of those plants where seed propagation is difficult. These buds may undergo a period of rest or dormancy, then it will undergo some physiological maturity before it is risen into a new plant. These bud dormancy can be studied under two parts: induction of bud dormancy and removal of bud dormancy. First, let us consider induction of bud dormancy. Resting buds are formed in woody trees, particularly of temperate region. Temperate trees form buds in late summer or autumn and resume growth only during spring. But the buds of the tropical trees resume growth without any resting period. Several physiological and morphological changes are responsible for induction of bud dormancy. Elongation of internodes and expansion of lamina are stopped. See, during the dormancy period, there is no growth, growth is reduced. Hence, the elongation of the internode or the growth of the lamina or leaf blade is halted. Size of the bud will increase due to meristematic activity in the bud and finally it may stop. Certain inhibitory chemicals may also be produced in the bud during dormancy, which will induce dormancy. Short day conditions may also induce dormancy in long day plants. For example, in Fagus sylvatica, Taxus baccata. Here, in many species, buds may resume growth after exposure to winter chilling. The effective temperature range to overcome dormancy is between 1 to 10 degrees centigrade. The length of the chilling period to break dormancy is 260 to 1000 hours. If previously I have told you about this Fagus sylvatica, which is a short day plant, but dormancy can be removed by transferring seedlings from short day to long day conditions. But dormancy can also be overcome by ethylene chlorhydrin, thiourea and dinitrophenol. It can also be overcome by treating with growth hormones like gibberellins. So, these are some of the things about bud dormancy. Now, let us start with seed dormancy. Seeds are very important from the point of view of our livelihood, agriculture, economy, everything. And this seed dormancy is a state at which metabolic activities of seeds are at low rate and germination is temporarily stopped. Also, it is called dormant stage or resting stage of any seed. Seeds at the state of dormancy are called dormant seeds or quiescent seeds. Fresh seeds undergo a period of rest for a considerable time, which is called dormancy period or period of dormancy. If these dormant seeds are sown in the field, even under favorable conditions, they fail to sprout or germinate. What are the physiological features of dormant seeds? Following are the physiological features of the dormant seeds. They will have very low moisture content, very low turgor pressure, and the reserve food in the endosperm or cotyledon will be in the insoluble form. The enzyme activity is at a very low rate, aerobic respiration is very slow, and there are presence of germination inhibitors in the seed coats. Because of the presence of these germination inhibitors in the seed coat, germination of many types of seeds are stopped, inducing dormancy. And there is a rate of metabolic activity which is very low. And what are the causes for seed dormancy? First one is water impermeability. This is the most important and widespread cause for seed dormancy. The hard seed coat and waxy coating of the seed are completely impermeable to water. There are many seeds like seeds of Malvaceae, Convolvulaceae. Here the germination begins after the microbial decomposition of the seed coat. Once the microbes decompose the seed coat, it becomes softened and then it can absorb water now. Next one is gas impermeability. Seed coats of certain seeds like xanthium are impermeable to oxygen. During dormancy, seed coat becomes gradually permeable to oxygen and start germination after the dormancy period. Next is mechanical resistance of seed coat to the growth of embryo. If anything, any hard and tough seed coat or seed coat is present outside the embryo, then embryo cannot expand due to growth. Hence, hard and tough seed coat, it's not growth, coat prevents the expansion or growth of embryo. Hence, seed coat is weakened gradually by saturation with water. Then the embryo grows to full size and germinates and thereby breaks the dormancy. Example is the seeds of amaranthus and capsula. If the seed coat remains dry, they are dormant even for more than 30 years. And this may be taken as an advantage feature for the research work, because we can store the seeds without germination for many years for the purpose of future research. Next is rudimentary and poorly developed embryo. Immature and rudimentary embryo may also be because of seed dormancy. During dormancy, the embryo gradually develops to attain full maturity and germinates. Example is ginkgo biloba. Need for after ripening in seed storage. The seeds of apple, pear contain mature embryo. They fail to germinate even if they are exposed to favorable environment. They will not germinate even after the removal of the seed coat. Such seed require some period of rest or dormancy. They need a period of dry storage to germinate. This period is called after ripening or period of after ripening. Next is germination inhibitors. Seeds of apple, rose, peach contain germination inhibitors like abscisic acid, ferulic acid, coumarin, etc., which prevent their germination immediately after the harvest. Such seeds require cold climate for germination, as low temperature has inhibitory effects on these. Hence, these seeds germinate in the spring because their low temperature treatment is fulfilled by the cold climate or low temperature of the environment. Next is light. Certain seeds are sensitive to light and they are called photoblastic seeds. Some seeds germinate after a brief exposure to light. They are called positively photoblastic seeds. These seeds remain dormant until they are exposed to light. Once they are exposed to light, they will germinate. Some seeds become dormant on exposure to light. They are called negatively photoblastic seeds. Resuming the germination potential of dormant seeds by special features is called breaking of seed dormancy. So, now let us discuss about breaking of seed dormancy, which are the methods which are used to break the dormancy. The seeds germinate on breaking the dormancy. There are many ways to break the dormancy. Methods of breaking seed dormancy. First one is scarification. It is just a process of mechanical breaking of the hard seed coat to break dormancy and is applied to those which have thick hard seed coat and impermeable to water and gases. It includes breaking or scratching of the seed coats mechanically to modify the hard seed coat or for easy permeability. Sandpapers are rubbed against the seeds to actually break the seed coat. Sandpapers may effectively, that's why sandpapers may be effectively used for this purpose. Next is methods of, the other method is hot water treatment. By soaking the seeds in hot water, it can break the seed dormancy. Seeds are soaked in hot water at about 25 degrees centigrade, maybe warm water 35 degrees centigrade for few minutes, and then it is soaked in cold water 10 to 15 degrees centigrade. This treatment softens the seed coat and removes the inhibitors. Example is Pova pratensis, Rumex crispus. Next is acid treatment. Soaking the seeds for a few minutes, that is 5 to 6 minutes, in concentrated sulfuric solution or hydrochloric acid modifies the hard impermeable seed coat. Then the seeds are washed thoroughly to remove any acid residue present there. Then it is washed thoroughly. The method is used for seeds with hard and tough seed coats. Seed stratification. Here the different layers or several layers of soil or soil-like material is used, that is stratification, different layers. The chilling treatment given to seeds by placing them in different layers of soil is called seed stratification. The treatment also removes the inhibitory substances. Seeds are spread in different layers, alternating with layers of peat moss or sand or any material of this kind. These materials are kept moist with water at a temperature of 5 to 10 degrees centigrade for few days. This process keeps the seeds away from light and at low temperature. Dormancy of light sensitive seeds can be broken by this method. Example is lettuce. Next is chemical treatment. By treating the seeds with chemicals like gibberellic acid, kinetins, auxins, these are all plant hormones, then nitrates, thiourea, dormancy can be removed. These substances promote germination. Dry storage. Storing dry seeds in dark and dry conditions actually promotes or break the dormancy. It promotes after ripening in certain seeds. It promotes after ripening period, which are the seeds which are dormant immediately after the harvest can germinate after they are subjected to after ripening period. So, the dry storage that we do it in the warehouses gives a very good method of breaking seed dormancy, because it provides after ripening period so that the embryo inside the seed will germinate, so that the embryo within the seed will germinate, or it will undergo a period of maturity and it can germinate. Next is light treatment. Dormancy of positively photoblastic seeds is removed by exposing the seeds to red light after soaking in water. The red light converts PR, that is phytochrome red or phytochrome red to PFR, and includes or induces the seeds to germinate. But if the seeds are treated with far red light, PFR is converted to PR and this inhibits germination. In negatively photoblastic seeds, the condition is reversed. And now let us consider the importance of dormancy. Dormancy is very important to nature as well as mankind. Seeds can be stored for future use for purposes like sowing, research, future propagation, etc. Seed dispersal or seed dispersal is brought about even during unfavorable conditions. Dormancy induced by the inhibitors present in the seed coats is highly useful in desert plants. Dormancy keeps the seeds and buds alive even during unfavorable conditions like extreme hot, cold or dry condition, as the metabolic rate is too low. Dormancy helps seeds to remain alive in the soil for several years and is a continuous source of new plants even when all the mature plants of the area have died due to natural calamities. It may also be a problem for farmers as they cannot sow the seeds immediately after the harvest, and they have to wait for sowing till the completion of the dormancy period. But this may be broken by the methods explained if required. Hence, the dormancy is a very important event in the life cycle of a plant species as well as for agriculturists and researchers. Thank you for listening to my lecture. Thank you.